Thank you everyone for having me. So today it'll be a switch from what we just saw, but actually it makes me rethink of some of the things I want to talk about here. So this is good. It's been very inspiring. The conference has been amazing so far. So today what I'm going to talk about I also slightly changed the title to be kind of unexpected, sometimes expected, challenges in using brain imaging to detect and treat psychiatric disorders. So what I really wanted to do here, so I'm a statistician by training, and the way that I like to think about things is trying to understand underlying uncertainty and how that can affect using models like large language models and these kind of other AI technologies that are being used so often. So I wanted to just kind of take that and put it into perspective of what we really have to think about and what the challenges are when you start to think about brain imaging. So let's see if this thing works. May not, but I can always do this. All right, so quickly a little bit about me. I like to always have a slide like this, even though Jim said some of it. I'm from North Carolina. Whoop, let me go over here. I met my wife at UNC Chapel Hill in grad school. She was also in statistics. We moved out to the Bay Area back when I graduated. And starting next year, I'm going to be the chair in the math, statistics, and data science department there. So that's, thank you. I appreciate that. And in 2021, I was actually a visiting assistant professor in the Department of Psychiatry at University of Pittsburgh as well. So a lot of this talk and some of the stuff I've been doing in brain imaging, as you'll see, has been inspired by a lot of the work I was doing there with psychiatrists. I'm trained as a statistician and data scientist with expertise in statistical network analysis. I'm going to talk about brain imaging data from multiple modalities, scanners, and studies today. And beyond that, I also think about social dynamics and influencers on social media, as well as interpretable machine learning. And so I think what you'll find out and what's kind of interesting about a lot of these brain data also is that the techniques that are used here can be used in many different areas. And so coming at it from a statistical network analysis standpoint, you can really see how actually social media kind of in some ways are highly related to what you do in brain imaging as well. So I've had some nice conversations about that. I also want to start, because I know there's a lot of students here, to just say my first experience with brain imaging data went horrible. I was a grad student, actually, so I was a PhD student, second year, and there was this study called ADHD 200, so I'll date myself a little bit. That was back in 2011. I was a second year naive PhD student, but I threw everything I had at this data set. So this was a competition to really see, can you use both imaging data as well as clinical information to try to predict different stages of ADHD. And so the outcome was healthy versus ADHD. It's still online for those that want to go check out that data set. It's a public data set that you can look at. It was a competition. I wasn't joining it from a competitive standpoint. But no matter what I tried as a second year PhD, I got nothing but absolute noise. Now, what was funny about that was I was always very interested in kind of diving into brain imaging data. But at the time, I didn't have the skills to be able to do so. So I've learned a lot since then. And I kind of want to share some of the information with you. So what exactly is brain imaging? It's a lot of things. I have two pictures here that show, an EEG machine, which is more of like a mask that is measuring electric current through the brain. But then you can also think about machines like this, which are for MRI or fMRI scans. And the scans can be structural or functional. And there's many different types to think about, and for many different reasons as well. And so the multimodal, I have to remember not to come to this slide. More than that, you can actually do multiple types of measurements at once and think about how you can combine these kinds of images. And so just as an example for a structural image on the right here, you can see this might be something that a radiologist looks at to kind of see if there's physical trauma or any anomalies in the brain. So what can we do with brain images? A lot. A very nice example that I like to think about is from Alzheimer's disease. And so particularly with Alzheimer's disease, it's one of the kind of rare situations where you can actually diagnose Alzheimer's disease through the use of imaging. And so the measurements of two different proteins are taken using PET, so positron emission tomography. And by looking at the concentrations of these two different proteins, you can determine whether or not someone has Alzheimer's disease. So that is the true and tried test to actually see for Alzheimer's. Now, it is also a vascular disease. And in that, there's a lot more tied to the structure and function of the brain as well that we can try to use for early detection. So there's a lot of opportunity there to kind of look for early detection as well. It's also used, and this is just kind of a list of ideas, and I won't go into it, but these are a lot of the different studies that I've also thought about and worked on. and some of them for treatment of late life depression, and I'll go into this in particular later. But then also relationships between sleep disruptions and stages of psychosis. And that might, I've been working with some collaborators at Pitt there with EEG data to try to understand this. And then also the development of schizophrenia and the stages of schizophrenia. I also wanted to say, funny enough, also political behavior. And I call this my 12 seconds of fame outside of this talk. But this talk is for another time. But this was on the, I'm proud of this moment, but this was like, this is what you saw. My paper is that one, I promise, but it's kind of behind the headline. But for using brain imaging to actually say something about political ideology as well. That was a fun study. But now kind of coming back for a second and thinking about the age of AI and where I want to go with this. So this is from a Nature paper in December of last year showing kind of a projection as to when it is we'll run out of data for these large language models, right? So, and I think people have heard this kind of problem, but LLMs, OpenAI, DeepSeek, all of these many different types of kind of artificial intelligence tools that are being developed very quickly. And I think Nathan said this very well. Part of that, of what you said, was more data equals better models, right? But at the end of the day, we do need to, if we put a little bit more thought behind the data that we have, we can actually get more out of it. And I think that's an incredibly important point, that we don't always need to just try to get all of the data. I'm going to talk about that as well. But at the moment, here's what's happening, right? So more and more of the data is being crawled online, and there's a projection of around 2028 that indeed will be out of data, right? It's using it so quickly. So what happens then? But my question is this: is more data better when it comes to brain scans? So what can we say about that kind of idea for brain imaging? Well, typical brain imaging studies are often small, and this is really due to costs. So the cost of an MRI or fMRI can be quite high. Now there are other more inexpensive scans, but sometimes they're more noisy, and there's a lot of considerations to think about there. But because of this, and especially in clinical trials where imaging, there are experimental components to looking at these images. So a major aim of current research is really to try to say, what if we could take all of these clinical trials, right? So they all collected data for various reasons and at various stages in the study, and try to somehow piece them together to get a more powerful representation of what's actually going on. So a really big component of this would be to say, hey, can we take all of these many different studies that the NIH has funded, say 100 scans for each one, and say something about the healthy trajectory of aging? Or what about for the development and detection of degenerative disease, right? So using more than just 10 to 20 or 100 scans. And stages of psychiatric disorders, all of these. And I will say there are large repositories, so large repositories exist. Some ones that are particularly interesting and have been around for quite some time include the ABCD study, the Human Connectome Project, and the UK Biobank, which are all somewhat publicly available. I say somewhat with an asterisk, like you have to ask nicely and then work hard to get the data in the right place. But at any rate, you can actually get access to it. So what could possibly go wrong? Well, the first thing that's incredibly important is the fact that individuals are variable. We're all different in some way for many reasons. And I give an example here just by looking at a very simple measurement, or three simple measurements actually. So for cortical gray matter, so just an amount, this is a structural scan, we might be considering, say cortical thickness, as well as kind of two other different measurements that we look through across time, but cortical thickness being a primary one here. And if we look across age, then we see, first of all, there's a trend, right? So there is a declining trend, which for those who study brain imaging, it kind of is scary, right? As we age, we see that our brain, in fact, loses mass, that the function becomes worse. So healthy aging is something that we do see it starting to deteriorate slowly. Sarah, you could also comment on that, I know, with the analysis she's done. But in addition to that, so we have this kind of general trend, but notice there is variability, right? So these have been measured as z-scores, and just trying to understand, well, look, we have a population at each one of these ages, but at the same time, they're not the same, right? There is variance. There is, what we could think of as possible stochasticity here. And so we've got to think about things like age, things like sex, race, health, stage of a disease. What does it mean to actually be healthy, and defining that? And these differences, by the way, so what makes this even harder is that these differences manifest differently across different brain regions. So that's kind of the other part of this plot, right? So we're looking not just at whole brain, so that's one aspect of this, but also according to different parts of the brain. So you can see how it's highlighted with the ribbon, frontal, parietal, temporal, and occipital lobes, and how we measure this. So these differ across these regions, but the variability differs across these regions. So being able to account for that is very specific to different anatomical regions, and we have to take that into account when we're thinking about these kinds of images. So the good thing is, though, this isn't an impossible question, and in fact, for those that like statistics, taking linear regression and things along these lines, you can imagine this to be kind of a regression problem, right? So it's really, well, if we have an idea of this, can we account for individual differences, by regressing out these confounding individual variables? I'll say the answer is kind of, right? And I'll talk more about that in the example that I give. But it's difficult to account for these individual differences over the entire topology of the brain. So the hard question, at what scale do we care about? Do we want to look at this at a very nuanced neuronal level? Do we want to look at this anatomically for different subregions in the brain? Or do we want to look at this at the whole brain level? And so these are all very interesting questions that we have to think about as well. The other question, or other point of variability to consider, is technical variability. And so I love this when I first kind of started thinking about technical variability. I love this plot because this is not exactly a realistic thing that happens in real life. What happens if someone gets an MRI? Most of the time, you go for one scan, or maybe you go for one scan over time, but generally speaking, you're going to the same scanner in some way. But what if you actually could go and get a scan done over many different scanners, right? And so that was the point of a study that I took part of as well, where we wanted to understand overall scanner variability. And so these are 4 different scans, each with the same resolution, same parameters set to give you a reasonable image here. And notice, so there's the same individual. Notice that the plots, those similar, so these are structural images, those similar are not the same, right? And in fact, below each one of them are distributions kind of the voxel intensity, so of the kind of pixel intensity of these scans. So the question would be, if I got my brain scanned under a GE scanner, okay, and David, who's coming next, got his done under a Philips scanner, how do we account for these differences, if we're looking for something specific? So that's, that's an interesting question. And so furthermore, in the case of wanting to add more data together, to aggregate data to get more powerful predictions and more powerful inference, how can we actually aggregate information across these studies? More practically, how do we compare downstream results from 2 studies that use different machines? There are some solutions to this, and a lot of people who are in brain imaging kind of do this automatically, right? So there's this whole pipeline of things that you follow whenever you're saying, I'm going to look across multiple studies, and harmonization is one of those steps, right? And if you don't do it, then your reviewer will certainly come back to you and say, what kind of harmonization did you do? I didn't see you write about it in your methodological part of your paper. But it's a very important part, because what this is trying to do is to account for these differences. And so harmonization as a technique is really just trying to remove non-biological inter and intra-scanner variability through some kind of normalization. There's really 2 main strategies for this. One, which is not harmonizing the images themselves, but rather trying to normalize over the measurements that come from the images. Okay, so you take the images first and you say, hey, I want to know something about white matter within a certain region. So you get a summary of this. And then what we do is do that across scans, say, and we'd like to normalize across scans to make sure that those summaries, those mean white matter intensity that we got, was comparable right across scans. And so that's the first type. And there's a really common method for that called combat harmonization. So if anyone in here had been working in the genome project and microarray analysis and things along these lines, combat actually comes back from the genome project in 2007, where it was first created then, over 8,000 citations to date, and now primarily being used kind of in the human connectome project. So to understand how do we normalize the different measurements that we can get from the brain. So that's one type. The problem though, well, hang on, I'll get back to pictures in a second. One of the problems though is that this kind of harmonization is prone to removing biological differences that are correlated with scanner effects. And it's hard to actually understand what the scanner is giving us individually, because we're just looking at a summary of these images. On the other hand, there's also direct, I call it direct harmonization or normalization of images. So we're directly going to take these images and try to map them to the same space, right? And so one such method that's pretty common is called RAVEL from 2016, and this was actually image normalization done at the voxel level. And I should say, I'm trying to remember exactly when it is that kind of style transfer in neural networks came out. Does anybody have a date for that? I should know that on the top of my head, kind of image transfer. Anyway, it was pretty recent. I would say it was after this. Was it after 2016? 2019, 2020. That's what I was thinking. Thank you. And so the idea since then, RAVEL has kind of become a little bit obsolete, as a lot of these things happen to be, right? Because of deep learning strategies that are now used to do this same kind of process. And so since about 2021, there's been many methods, unsupervised models used to treat harmonization as a style transfer learning problem. It requires a target scanner, though. So if we have multiple scanners, we have kind of this target image that we'd like to map everything to be in the same space. And so this example, you don't need to follow the entire map, but what I want to say is it's an example of kind of what you'd expect to see with something like a combat harmonization. And so this one's called, and by the way, these three types of harmonization methods I've also worked on with collaborators. And it's kind of taking combat and extending it in some way. Not all of them, I'll show you a different one in a second. But this one is taking combat and trying to say, rather than simply looking at variables that come directly from the image, we're going to actually break down the image itself into what we call superpixels, and then understand measures at the superpixel level, so that we're getting a little bit more of a granular understanding of what's happening in the image. So this was recently published in Alzheimer's and Dementia. And the idea was it was used on this data set that I was looking at before for kind of understanding different or brain differences and normalizing data across people who had 4 scans, and each of them were in stages of Alzheimer's. And so what you can expect, just to give you a kind of an overview of this, is you start with the raw images. We then do pre-processing. Now, pre-processing, I could also call that an issue with brain imaging. There's a whole massive pipeline of pre-processing. And anyone who does brain imaging data analysis, at some point will have to learn how to pre-process, unless we can just get a deep learner to do that whole process for us. That's really what I would like. So I think that would be an amazing thing to do. But there's a lot of steps. And as a statistician, I've gone through and tried to understand most of those steps. I can't say I understand every single one of the steps. But it is one of these things that, there's a lot of choices made to get from a brain image to the normalized brain image, right? So I'll leave it at that, just to say there's a lot of different things you could try. And whenever you read a different brain imaging study, there's always this section that's saying, what's the pre-processing, right? How do you get through that? And if you notice, there's a lot of variability there. So it's kind of what the practitioners typically like to use. So, and registration to a certain space. And then the idea combat is finally happening. So it's taking these 4 different images that are now kind of registered, so they're comparable at least through rotations, to think about different effects, so additive effects of scanners and multiplicative effects of scanners, and then basically trying to regress that out. And we're doing this at a superpixel level. And what that ends up giving you are different maps that are saying what happens whenever we start to normalize based on these now superpixels, or these maps, which in this paper we called the gamma, delta, mean, and variance map. So we looked at 4 different maps. And then average out over these different maps to finally apply that back to the original data and get, voila, harmonized images that now have a similar distribution of these different voxel intensities. So the thing I want you to keep in mind is essentially what we're looking for here. These all images were turned in the proper direction, and acceptable in terms of how we're looking at it, not too much motion and many other things that we consider. What we'd really like to do is make all of these distributions look as close as possible to the same while maintaining the integrity of the information, right, the biological information that each scan is giving us. That's what we're aiming for. And so these harmonization methods are trying to do that, right? Some other, and I'll just briefly mention this also to make sure we stay on time, so I won't try to take up too much time either. But for, interestingly enough, something I've found, so being that I'm a networks person, I said to myself, why hasn't someone tried to harmonize while maintaining some form of structural connectivity? So this is actually a paper that's under revision right now. We just sent in, actually yesterday, during this conference, our revision to neuroimage. So here's hoping, my fingers are crossed. But the basic idea is similar. It's still using a combat-based model, but now thinking about kind of parameter estimates that are coming from either a block model or some form of network model that describes the underlying connectivity. So again, hopefully that comes through. But it's another way of thinking about this. And then finally, a different kind of method to show that's possible here is, in fact, and I believe this has the encoding and decoding, so I'm going to consider how to get rid of the inverse step based on Nathan's talk, so thank you for that. But this paper's already been published, but I'd like to investigate that further. But the basic idea here is to actually directly map images, right, to harmonized slices. So by considering, since we have in our example 4 different scans that we're looking at across many people, kind of bringing them all to a middle ground harmonized space. So there's no ground truth, but we do have kind of matched data. So we're able to use the relationships between these scans to understand what an average should look like, right? And we're just trying to embed all of the images to be in that space. So it worked incredibly well in this application. And so as you can see, this is just a simple example of the application, but these distributions that we had for intensities over each image, we ended up getting back to a distribution that's quite comparable across these 4 different scanners. And then what we'd want to do if we wanted to aggregate the data for each one of these scans is indeed think about, what the new images look like that have these kind of normalized or now similar distribution images. And so key takeaway, there is hope, right? So we can actually do something here. We can transform these distributions, but it is a complicated process to be able to do that. But now what I do, so when I'm talking about possible issues, so some of these, and I said at the beginning, some of these are expected, whereas some are unexpected. And so if you read enough and kind of or get into the brain imaging literature and imaging in general, medical imaging, then you realize that these types of variability are quite expected, right? So this is something that we kind of have thought about for a while and that we can try to handle, right? So people come up with solutions for this to see what we can do. But now I want to actually go to a case study that shows a little bit more of a subtle example of something we need to think about for these kinds of images as well. So I'm changing gears a little bit, and I actually want to discuss a case study which is the treatment of late life depression. And so this was also a collaboration that I had with with folks looking at who are in the ADRC at University of Pittsburgh. And so here, just to kind of give motivation, major depressive disorder, MDD, is among one of the most common psychiatric disorders. Around 300 million people globally have MDD. It's the 3rd highest disability adjusted life years of any cause, and the highest disability adjusted life years of psychiatric disorders. So it has a massive impact. And then economically, it's about a $210 billion burden. So thinking about this is incredibly important, but also a very burdensome disorder that we need to think about. And so in particular, what we've been looking at is late life depression, which is generally thinking in geriatrics for people who have major depressive disorder. And so one thing to think about, a very common mode of treatment is to use an antidepressant. And there's several forms of antidepressants that we could think about. And generally speaking, the way that this works is someone goes in, they get a clinical assessment. That clinical assessment kind of determines, based on conversation as well as a survey, and I'll talk about the survey in a second, where you are in a certain scale. So there's kind of clinical scales that determine whether or not you have depression and to what extent. And from that, a certain type of pharmacological treatment is given. Sometimes not pharmacological, but I'm thinking about pharmacological. So some form of treatment is given, an antidepressant. The problem is that it generally, before you can actually understand whether or not that treatment works, takes at least 3 weeks. So you're going back to the clinician, you're still talking to the clinician, and they're trying to understand where you are now in this survey of kind of how depressed you're feeling, whether or not it's working. But to actually understand is generally about 3 weeks. And so not only that, but less than 50% of people who take the antidepressant actually respond to the first line of treatment. So what does that mean? That means, 3 or more weeks of unsuccessful treatment can lead to bad, often fatal consequences. So we're talking about people with major depressive disorder. And so that's a really big issue. So there's a very important kind of prediction task here, right? So when we're thinking about machine learning, here's a really good one. Can we make these predictions of whether or not someone will successfully, if a first line of defense, a first line of treatment will successfully treat someone? That would be amazing. I mean, this would be an incredible opportunity for machine learning and things along these lines. And so that's the goal of this work. So can we actually find more efficient ways of predicting whether these antidepressant treatments will succeed for an individual by augmenting clinical assessments with brain imaging? Well, I wouldn't continue talking about this if we couldn't do something. So the talk is not over yet. So let's go into it. What can we do? So first of all, the gold standard, I wanted to say, for clinical assessment of MDD is what's called the MADRS score, the Montgomery-Åsberg Depression Rating Scale. It's a 10-question questionnaire given by a clinician that assesses sadness, reduced sleep, reduced appetite, pessimism, as well as suicidal thoughts. So I know some of us in here have a newborn baby, and I know reduced sleep, but we could talk about other types of scales as well. But each item has a scale of 0 to 6, right? So 6 meaning the higher severity. So total score of assessment. Now these, you could always talk about how do we come up with these thresholds, and that's a whole other question, but through evidence. So no or minimal is 0 through 6, mild depression 7 through 19, moderate depression 20 to 34, and then severe depression 35 to 60 is typically how these things are treated. So we have a case study where actually it's 2 different studies. The one where I came in on this was actually what's called NEMO. Now they came up with wonderful names. I'm terrible with acronyms, and I feel like you have to be good with acronyms to like SHRED. That was wonderful. But I'm no good at that with algorithms. I got to get better at that. But NEMO was the name of the study, and Circuits 2 was another one, and that was from 2016. But the idea here is we were looking at 2 different populations. So the reason why we're looking at 2 different studies, one is to kind of understand this idea of having higher N. We wanted more people, right? We wanted to understand, can we do this across 2 different studies and actually get robust results? So that was the other thing. So kind of reproducibility, or the fact that, test, retest, in the sense that where you can use NEMO and see whether or not that works in the Circuits 2 data and vice versa. So they were both taken by this same group at the University of Pittsburgh. And the NEMO study, age 55 and over, at the beginning, so to be enrolled in the study, your MADRS score was greater than 15. There's also another mini mental state exam that I did not talk about, but this is another way of scoring a kind of awareness of where you are on mental state. And so whenever that score was greater than 21, there were different treatments as well. So I should say, over here there's venlafaxine, and then in this study there was Fatsima and Lexapro, split up accordingly. And then N equals 51 in one state in the NEMO study, N equals 29 in the Circuits 2 study. And they were both done with, we got resting state fMRI, was the kind of imaging we're going to be looking at, with a 3T scanner. So here's the idea. It was a 12-week study. Both of them were 12-week studies. And they were conducted in the exact same way, where to begin with, on the very first day, the person would come in, get an MRI, so a structural MRI as well as an fMRI scan done. And they were given a placebo on the first day. And then day 2, they would come in, and we'd get their scan, so do an fMRI scan. And this is actually what we're going to call our baseline scan. So they kind of had already come in, taken a placebo, and then we measure what happened, or like where their brain, functional connectivity or functional MRI at that time. And then in day 2 at around 2 p.m. is when the first medication, the actual medication, is given. And that's kind of the start of the treatment trial. So then about 12 to 18 hours later, they got another fMRI. All right, and we're calling that kind of the day 1 scan, not to be confused with the fact it's actually day 3, but we're looking at baseline and then 1 day after, right? So this is about 12 to 18 hours after the initial treatment. And then there was another fMRI at day 7, and then finally on week 12, so it's 12 weeks long, there was yet another fMRI and MRI scan. So you can see this is, pretty difficult on the patients in a lot of ways. They're getting a lot of scans, right? So that's something. I mean, it's kind of it's a rare data set in that sense. It's pretty hard to get these kinds of scans over and over again where people actually say, yes, sure, I'll do that, during the study. Furthermore, the clinical assessments were taken about 1 hour after each brain scan, and the MADRS scores were taken every week. So they were being evaluated every week to see where they were on that scale as well. All right, so outcomes of interest. So the main outcome of interest that we're thinking about is what we're calling remission. And that is just a binary outcome of whether or not the person had a final MADRS score of 10 or less for 2+ weeks. Okay? So they were followed up with, still got another clinical assessment, just making sure that their MADRS score was still below 10 for the use of the treatment. And so predictors, we had demographic information, so age, sex, race, education level. We had clinical predictors as well. So there's another measure, the Cumulative Illness Rating Scale Geriatric, so it's called the Sears-G. HAR scores, which are measurements of anxiety, and then there's baseline MADRS, were the other kinds of predictors we were looking at. So kind of a baseline understanding of the scale of depression. Typical predictive models. So generally speaking, what people have done in the past, without having kind of fMRI information available to them, has been to say, well, we have this clinical information, we have demographic information, let's fit our best possible binary classification model, right? So binary classification models for remission, right, to try to understand this. You could use something as simple as logistic regression. You could use random forests. Use your favorite option. Most of you have had some form of a supervised machine learning class. There's lots of options. Logistic regression and random forests are typically pretty good places to start. Using the above kind of predictors, and the best performance in the literature was around a 0.67 AUC. So people have been looking at this and saying, okay, well, we can predict reasonably well. This is much better than random chance. So that's nice. But what we would like to do is try to build a more predictive model of this, of remission, using functional connectivity networks from fMRI. To kind of give you a brief understanding of this, so fMRI is measuring, it results in a time series, results in a time series where across each kind of voxel or small cube in the brain, we're getting a measurement of bold response, which is blood oxygen level dependence in that region at every given moment in time, so being measured repeatedly. And so we get that for each voxel. And generally speaking, for for very high resolution scans, sometimes this can be hundreds of thousands up to 1 million different scans that we're getting, voxels that we're measuring over. And so we're getting this really large kind of multivariate time series where we're looking at kind of voxel bold response over time. The way that we think about functional connectivity, which is a lot of what my work has been kind of focused on, is to say, well, let's build a functional network out of this. And so what we get are fMRI snapshots kind of at each, say, interval in this time period, over the time that they're under the machine. We then get from that a multivariate time series, and basically by looking at the multivariate time series, we calculate some form of co-activation, usually correlation, something like that. There's a lot of choices there as well. But for simplicity, we'll talk about correlations across the regions of the brain. And that's our functional connectivity matrix. So we try to say, let's put edges between things that are highly correlated or tend to co-activate at the same time. So this is kind of a representation of how function, or communication, kind of function co-activations within the brain. So why network analysis? I think I'm going to skip that, but I'm just going to say, I'll say the last point, because I could talk about networks all day and I don't want to get too overly excited about this, except to say the following thing, that my collaborators there had tried looking at baseline and day one functional connectivity matrices and had 2 published papers on this idea for the circuits 2 data, and found in fact that you can get an increase over the kind of classic clinical models by around 15%. So get an AUC up to about 0.75. So I thought to myself, well, this person who did this is not a networks person. I was trained as a networks person. I'm going to put at this what I know from network analysis and see what I can do, right? So that was my hope. So can I use the skills I now have in network analysis to get there? And so what I ended up using was this thing called differential connectivity, which still, for those that do network analysis, understand this is a very simple measure. Like, I didn't do anything super special here. I tried many other things, by the way. I'm just not putting them in the slides. But here's what I want to point out. So I actually looked at So I wanted to point out, so the previous analyses was baseline and day one functional connectivity. We had functional connectivity for many, many other days, right? So for many other weeks after the fact. That's still unexplored. So there's a lot more exploration we could do. But keep in mind that the whole aim is to try to come up with as early as possible a prediction for remission, right? So we're saying, can we do it in day one? And one of the clinicians that's on the study was like, we can do it in 24 hours. So I stood by her. I was like, let's do this, all right? So I'm trying my best. I'm doing some kind of differential, saying how did your functional connectivity change at day one from baseline after you took the treatment? That was my goal. So we have pre-processing and everything going on here, functional connectivity matrix at day 0, at day 1, and then we get overall DC, which is just taking day 1 and subtracting day 0. So very simple difference here. And that ends up being, we're looking at 3,321 pairwise connections. All right, so we're looking at basically edges and the differences between them. And so this can be positive or negative, right, in what we're looking at. And then there's exploratory analysis, which is kind of my favorite part of statistics. And then there's predictive analysis. So we built a random forest model here using now these 3,321 different pairwise connections in addition to all the clinical information and demographic information that we had. All right, so now drum roll, how did we do? Not well, actually. So I was really bummed by this, but I also wanted to point this out as part of the data science and artificial intelligence kind of system, right? Part of how we learn things is by failing first. So what I want to point out is the following thing. And then I'll tell you why it happened, and this is the subtle aspect of what I learned. So initial model, so I put this at the top, but then I kind of give away what I'm about to say. I'm not good at like putting two different parts in a slide. I got to get better at that. But what I want to say, initial models did not improve predictions from previous circuit 2 analysis. Now, one might say, well, now you're actually combining across two different studies. There's inter-study variability. I tried to handle that using something like Combat. There's a lot of things you can talk about there. But actually, I didn't just try those edge differences. I was also trying motifs. I was trying all the network statistics, things that I know, throwing it at it. Couldn't really get anything that improved upon it. And then I started to think about it. I was like, I've got to start from scratch here a little bit. I was like, the network should be working. Like, I know that there's an advantage here. So what can I figure out? And doing more research on this, and then kind of diving in, and I just said, I'm going to try something, which is to divide the data. And I kind of think back to so much of medicine is done without really thinking about kind of sex-specific differences, right? And so this kind of was getting my attention. It was from other talks that I'd seen, but it was also from just seeing a lot of people doing amazing things with kind of what happens when you consider men and women separately in medicine and, better results, so the better results that you get. And so I did this. I said, okay, let's break this down. So let's look at the differential connectivity for a second. And what we ended up finding, so this is an image that shows these DC connections, right? So the differential connectivity in both males and females. And I looked, these are just plots to show when did remitters, so there's kind of two things going on here. So there are remitters versus non-remitters. So we wanted to say, how do remitters compare to non-remitters? Like, how well can we try to classify this data using DC information? That's our hope, right? So in doing that, we're going to say, well, let's look at the differences in remitters. Here's the positive difference. Here's the negative difference, right? And we did that both for males and females. And what I want to say is that these DC differences, overall and regional, were very different. So males and females responded incredibly differently as far as their differential connectivity goes. So the way that that brains were changing from baseline to day 1 and looking region to region, we saw an immense difference in kind of where there was greater differential connectivity versus smaller differential connectivity. There was overlap as well, but there's a lot of differences. So that was the first kind of point that I noticed. But then, of course, I really care about that for prediction, right? So our whole aim is prediction of treatment, like of whether or not treatment will be successful. So following suit with this, so my first analysis is some form of inference, right? That's classic statistician in me. So these were T-stat maps. And then in looking at this, I'm saying, well, you know what? I'm running a random forest. I can look at the variable importance of these different DC connections one by one and look at them in terms of how important they are for the prediction of remission. And so for each one of these, we normalized the variable importance. We ran Monte Carlo simulations to get some form of statistical significance to try to understand that. And the values here were derived from random forests. And again, when we're looking at importance, comparing males to females, completely different. Like there was almost no overlap in terms of where regions of the brain that were really important for prediction of remission in males were very different from those in females. And here's kind of a difference of these two plots for variable importance. So that was exciting. I was like, okay, so something's going on here, right? So why isn't my model working? Maybe it's simply because I'm not accounting for sex. You know, I regressed on it, and this is kind of the subtle point I want to make. So you might think, why didn't just putting the variable of sex into the model work? Like one would think that would. I'm claiming that this is too complex of a system to just put in a single confounder. There's actually interactions going on here, right? Based on the spatial connectivity, the actual connectivity in each person as well. So we tried many different models. So this is kind of my other way of thinking about overall analyses, right? As I say, well, there's a lot of different models we could try. Let's try them all and see what happens. So I have models A through G, but I'll show a point here, you know, containing different subsets of the following types of variables. So the first is demographic information. Next is clinical. The next, depression symptomology. Baseline connectivity, so the baseline connectivity by itself, and then also differential connectivity. And so I was trying many of these different models, and you can see accordingly which one is where, but I'll show you. Then I built predictive performance. And again, this is across two studies to kind of understand robustness. And there's a couple of, this plot is very interesting to me because what I'm showing are actually three different results. So there's three models for all of these models, right? So talking about someone who likes fitting a lot of models, this is what I like to do in my life and compare them all. The red or pinkish color is whenever we put males and females together. So we fit the random forest, we got our best predictor, I get an error bar on that to understand AUC. And then the green bar, the middle one, is always going to be just females only. So here's, I put females into the model. By the way, note, power's going down, right? So the power of this, it should be getting worse, like in some ways. If we're just thinking about this statistically, if males and females didn't matter, we should be doing worse with smaller samples, right? So that's kind of the interesting point here. AUC is going up. And then if we do male only, all right, so blue. Now, it differs across, but what I find here is actually in many of these different, and actually most of them, except for model E, which model E, what was it? So in all models but model E, which did not contain differential connectivity, that was the only one that did not contain differential connectivity, we see there were statistical differences in these AUCs, the predictive performance, between at least one of female only and all, and male only and all. So there was an advantage in prediction by actually separating the data. Okay. So I can kind of end on that because it's a subtle point, but I think it's a very important one and something I'm kind of creating a name for. So you're the first ones to see it. I'm going to call it the wrong data problem. So I actually think the wrong data problem is real. While it is useful to have more information, right, and statistically we could talk about that as well, about an outcome we would like to predict, it is wrong to believe that all available data should be used in a problem, or that having more data will necessarily give better results, okay? So in fact, when we're looking at this, it took going deeper into the data, understanding what's going on, trying to separate and look at these really complex models by separating males from females to actually understand how to get a better predictor. And we couldn't have done that by combining them. So issues like individual variability, technical variability, and intrinsic biological differences make it difficult to simultaneously analyze large numbers of brain scans, though there are some solutions. So we must understand and compensate for possible differences in the data obtained from disparate sources, technologies, or people. So I say, though, there is hope, but thorough and in-depth analyses are required. All right, I can end there. So thank you. Yes?
I was curious, so the MADRS score less than 10 is your outcome, right? Is whatever for remission. So Did you, so the makeup of that score involves multiple subdomains that may not move in a correlated way. So did you consider anything like an item response theory kind of strategy for evaluating your network connectivity and the different subdomains?
That's a great idea. We did not. We kept it a pretty blunt measure, to be honest with you. But I like that idea, to think, okay, what are each of these separate questions telling us about depression, right, or about the state of the person? Wonderful idea. Something to look into, for sure.
And really, really interesting, and I think that whole point about having the right data, and you know, the Google paper a few years ago on data cascade failures.
Exactly.
You know, having that knowledge.
Exactly. Thank you. Thank you.
All right, so I know you talked about some, but is there a way that you could have just clustered your DC data to begin with and gotten out the two subgroups that way so that you knew they were there initially, or was it more subtle than that and it would have been clustered? It looks like you didn't try it.
No, it's a good question. Actually, and that's really funny because often I'm like, I first try clustering and see what we can do, right? So I think interestingly there, I did not, although having said that, we talked about clustering networks, right? Relatively new area. I have not really delved into it. I think that's a very interesting and useful idea. It would be cool to see how well they cluster because I imagine it wouldn't be perfectly aligned with males and females.
We've looked at HCP data and you can see clusters that aren't perfect but correlate male fields to female some.
Interesting.
And so that's why I'm curious. But because I could also see it being subtle enough that maybe clustering wouldn't distinguish it.
Right, right. I will do that as well. You guys are giving me great ideas. Thank you. No, because that'd be interesting to see how well they align. Yeah. All right, great.
Okay, thank you.
Thank you.
For those of you who could not catch the entire session because of the second monitor not cooperating with us, we do have a 4K recording which will be on some digital platform and we will share the link with you via email.